The lab this week is on life chemistry. It is divided into 5 different sections. You will need to work through all of them. The first section doesn't have any questions that you have to answer over it. It's primarily a review of what elements and atoms are, but you should go through that because it'll help you as you answer the further questions in sections 2 and 3. Section 2 gets into molecules. And you'll see these pictures that are representations of different molecules. Each of the little balls, the colored balls, represent a different type of atom. This is a water molecule. The yellow balls are hydrogen, and the red ones are oxygen. So water is H2O, 2 hydrogens bonded to 1 oxygen. Here's an ethyl alcohol molecule. It has black in it. The black ball represents carbon. So there's a little guide here that tells you what the different colors mean, and also this is in your lab manual as well, so that you can answer the following questions, which have to do with bonds that form. Remember, when we're talking about bonding between atoms, we're discussing the electrons that are either being shared or exchanged between atoms, and every atom wants to have its full complement of 8 electrons in its outer valence shell. So it will borrow or steal or share electrons to get to that full complement. So we're going for a total of 8. Oxygen, shown here in the red, has two bonds or 2 electrons that are available for sharing. So it'll form typically 2 bonds at a time. Carbon, let's go back here. Carbon, which is black, it has 4 electrons in its outer valence shell, and it really wants a total of 8. So it has 4 that are available for sharing, so it can form up to 4 bonds. Looking at these 2 pictures, these diagrams will show you how that happens. So here's a black carbon molecule. It has 1 bond to an oxygen, another bond to another carbon, and then a double bond to another oxygen over here. So this black carbon atom has formed 4 bonds. 1, 2, 3, 4. So that's the maximum that it can form. Hydrogen forms 1 bond, so you'll see each of the yellow hydrogens is only going to be attached by 1 bond to the different atoms. Looking at nitrogen, which is the blue one, you'll see that it has 3 bonds. So it has a bond to a carbon and then 2 hydrogens bonded over here. So you can use that information that you just did to answer these questions. How many hydrogen atoms can be bonded with oxygen? We know that oxygen can form 2 bonds, so therefore 2 hydrogen atoms can be bonded with oxygen because each hydrogen atom is basically 1 bond apiece. How many hydrogen atoms can be bonded with carbon? Well, carbon can form 4 bonds total. If each hydrogen atom is equivalent with 1 bond, then that's a total of 4. And then a carbon dioxide molecule, which is a carbon with 2 oxygens, will have these double bonds that form. So 1 carbon is bonded to 2 oxygens, but there are double bonds that have formed between the atoms. So in a carbon dioxide molecule, you have 2 oxygen atoms bonded to 1 carbon, and the bonds are covalent bonds, double covalent bonds. The next thing you're going to do is you're going to look at several different pictures and identify the different molecules that are present or the different atoms that are present in each molecule. This first one is glucose, which is a very important molecule. It's what the cells will use for fuel. They will convert glucose into energy in the form of ATP, which we'll discuss when we get into cellular respiration. So you'll count the number of carbons, hydrogens, and oxygens in this molecule and record that in your lab report. The next molecule is DNA, which is quite complex. You'll see that it's double-stranded. It's made up of 2 strands that wind together. And so we call that a double helix. And that's the term used for DNA, is that it's a double helix and it is a spiral-shaped molecule. When you're looking at the different types of atoms in the molecule, you'll see another one, another little white molecule. This is, or another white atom. This is phosphate. So when you're filling out your answer down here for the different types of atoms that are in DNA, you're going to have hydrogen, oxygen, carbon, nitrogen, and phosphate. So the hydrogens are located here. We've got our black carbons, the blue nitrogens, red oxygens, and then the little phosphate ones as well. The next section, you're going to be identifying, based off of these molecular formulas that are provided, you're going to look at the pictures here and give us the molecular name. So looking at this first one, CO2, let's look over here. CO2 looks like this. It's the carbon with the double bonds to oxygen, and that's called carbon dioxide. So you'll record carbon dioxide in your lab report. Let's look at the second one, C6H12O6. Well, that's down here, C6H12O6. Here's the structure. You can see both the 3D and the organic chemistry form of the structure. It's a ring. And the name of that is glucose. So that's what you'll do to fill out the rest of this table. And then you're going to move on to section 3, which has to do with testing for different compounds in solution. So you're doing tests for carbohydrates, lipids, and proteins in solution. I did write down some information in the helpful hints section that goes over testing and controls and the different positive and negative colors for these solutions. Make sure you read that before you get into this testing. It'll make it a little bit simpler and make more sense if you know ahead of time what to expect. But basically you're going to work your way through different tests to test for different substances. Lugol's iodine solution tests for starch. And you'll watch a video. Benedict solution tests for sugar. And there'll be a video on that. And then Sudan-4 is a dye that stains lipids. You'll watch a video on that. And then Biuret solution tests for protein. And so it turns this purpley blue color if there's a positive for protein in the solution. So you watch a video on that. So this is our summary table. You have the type of molecule that you're testing, the reagent that's used to test for that, or stain it in the case of Sudan-4, the sample that we tested, what a positive color is, and what a negative color is. And that's the information you need to use to answer these questions under the different tests. And you're going to be recording the results of those experiments that you watched in the video under the table 3.3 below. Now this part, testing food samples for the presence of starch, sugar, lipids, and proteins, that's going to be this table right here. You're going to have 2 unknown food samples and you're going to perform all 4 tests on them, and you'll record whether they're positive or negative for that test, and that's going to help you figure out what these food samples are made up of. And here's pictures that will show each of these things. So the blue-black color is a positive for starch on this first test.
Locate starch in potato cells.
And then the last little bit is going to be a video that you watch that has to do with locating where the starch is actually found in the potato cells. And under this 4th question, you don't have to draw the diagram in the lab report itself, you just draw it in your lab manual. The next section has to do with molecular size, so that's section 4. And you're going to watch several videos that talk you through how we can use dialysis tubing to separate molecules based on size. So dialysis tubing has tiny, tiny holes that will allow small molecules like water through, but it will not allow the larger organic molecules to slide through. So it's a differentiable, permeable membrane. Some molecules can go through and some can't. So these videos will walk you through that so that you can answer the questions under these molecular size tables and questions here on the lab report. And then the last section has to do with pH measurements. When you're talking about pH, you're talking about how acidic or how alkaline or basic a compound is. Neutral on the scale is considered 7. The scale ranges from 1 to 14 or 0 to 14. 7 is neutral. The lower the number is, the more acidic it gets. So notice that lime juice and stomach acid are considered 1, which is extremely acidic. The higher the number, the more basic or alkaline it is. You'll notice that milk of magnesia, ammonia, all of those are considered very basic solutions. And you can use the scale to compare the relative acidity and basicity of different compounds. So you'll be running some tests with these different solutions on the video, and you're going to record the results of those tests on your chart and discuss the pH, whether it's an acid, base, or neutral, and then you'll answer questions related to that. The last bit is the summary questions. Don't forget to do those, and it'll just go over the important concepts from the lab itself. If you have any questions about the lab or if you run into any problems as you're working through it, please don't hesitate to email me.